What's up, guys? Welcome back to How to Tissue Culture. My name is Kevin Hernandez, and I love plant science, and I love to do tissue culture. And in today's video, we're going to be talking about the 5 stages. Usually people talk about 4, but stage 0 is very important. So we're going to go over the 5 stages of plant tissue culture, and hopefully this can teach you something, or it can motivate you to try it out yourself. So hope you enjoy the video. Alright guys, so to start us off, we got to talk about stage 0. So this stage is often very much overlooked, but that ends up being a problem down the line, especially when you want clean materials, clean plants. And so at this point, your plant material is being grown out normally, which is in a greenhouse, out in the field, out anywhere, not in an aseptic vessel. The most important part about this stage is to get your plant ready for tissue culture. And sometimes people think that you can jump straight into stage 1, but that often leaves a problem. Too many plants are contaminated, too many plants are too dirty, especially if you're taking plants that are grown externally outside, and sometimes even in greenhouses. Ideally, grow chambers that are basically completely controlled environmentally are ideal, but otherwise that's not always the case. You're not always going to have ideal conditions. So one of the ways you can take advantage of stage 0 is by taking cuttings of whatever plant you want and propagating them in a cleaner environment, whether that's going from outside into a greenhouse or from a greenhouse into an indoor grow environment that is completely sealed off. So a grow chamber, a grow tent, something that's a bit more manageable and disease-free. If you can do that, then you're already doing stage 0 and you're off to a great start. Alright guys, moving right along to stage 1. So now, stage 1 is the second stage, but people overlook stage 0. Anyways, stage 1 is the next step after stage 0, right? And in stage 1, you are going to be focusing on 3 things: surface sterilization, cutting your plant material to the right size or the right parts of the plant that you want to use, and 3, putting your plants into nutrient-rich agar in an aseptic vessel. I made this gesture because usually I use the square box magenta GA7s, but you can also use baby food jars, you can use test tubes, you can even do petri dishes, wherever you would dispense your clean agar into. And so you got to surface sterilize it. Usually people use chemicals or heat. I sometimes like to use bleach, a solution of bleach and a solution of ethanol. Sometimes I use isopropyl alcohol. So it all depends what species you're working with and what exact tissue you're working with, whether it's leaf tissue, stems, roots, nodal tissue, whatever. So that's stage 1. It's the cleaning stage and the plant preparation stage for tissue culture. Stage 2. Alright, so now we're going to talk about stage 2. And in stage 2 of plant tissue culture, you're going to really focus on multiplication and subculturing. And so what you're going to do now is create media that has plant growth regulators inside of it, PGRs. PGRs are basically phytohormones or plant hormones, and all they do in this case, we're going to be focusing on using cytokinins. There's 5 main categories of plant hormones, and we'll talk about those in another video. But stage 2, essentially, you want to use cytokinins, which will stimulate the top growth of your plant, create more shoots, create more plant tissue, and allow you to do the other part of stage 2, which is subculturing. And you're going to subculture your plant material as it grows, as it gets older, and as it needs fresh media. This can be done anywhere between 2 to 4 weeks. Sometimes people push it to 6 weeks. It all depends how your plant's doing, and you got to pay attention to your species. Some plants react a little faster, a little slower. There's important things to notice, though. More plant hormones, more PGRs is not necessarily better. Sounds better because, humans, more is better. But the problem is that if you use too much PGRs, if you do too much, it can cause side effects. It can cause not worthy effects that you would want to avoid. So whatever you're doing, if you're going to use PGRs, make sure you're using the right amount and the right type. But we'll go over that in much more detail in another video. Alright guys, and so once you're done with the shoot proliferation stage, which is stage 2, you're going to move on to stage 3. And on stage 3, you're going to focus on root development and root formation. And essentially, how are you going to do this? PGRs. Again, plant hormones. And so now you're not going to use cytokinin. You're going to use auxin. Auxin is the type of hormone that produces and encourages root growth and development. And at this stage, your plantlets, all your plant vessels, are going to be developing roots according to how they usually grow, but can be stimulated by auxins. And again, it's very important to notice, more is not better. It's like expecting to put more gas in your car after it's full and thinking that that's going to make it run faster or have more horsepower. It's going to make it go, but it won't make it better. So that's the kind of mentality you want. It's also going to cost more if you use more, right? So stage 3, root development. Alright guys, so if you made it this far into the video, we are basically at the last stage. And so now you have your plant grown, it's rooted, and now it's ready to be grown outside of tissue culture, taken out to the field, the greenhouse, or whatever you're going to grow into. But how do you do it? So in stage 4 is the acclimatization stage, which means you're going to get your plant slowly transitioned from an aseptic environment into a normal septic environment, or just growing out in soil and whatever substrate you're going to use. And basically that is the slow transition between a high humidity protected environment into a less humid, more normal condition environment. So as long as you make that transition slow and you possibly take your plant from the growing vessel into a humidity dome and then eventually into a normal growing environment where it's a lot less humid, you should be fine. And so the biggest problem with this is that people try to rush their plants, or they don't give their plants enough time to acclimate, and they die off, they dry out. So make sure to give them time. Take it easy. Be good to your plants. It's a lot of work. Alright guys, and those are the 5 stages of plant tissue culture. If you can do all those 5 successfully, then you can plant tissue culture yourself. And I will make a video on each one of the stages, going into more depth and going into more pros and cons and tips and tricks. I'm definitely going to make another video on PGRs and talk about the plant growth regulators in the future. So please stick around, and if you like this video, go ahead and drop a like, comment, subscribe. I'm very new to making content and just being in front of the camera, so this is weird for me, but there's a lot of fun things going on in the lab, at work, and some good experiments and good experimental ideas that I have planned for this channel. So if you want to support me, support the channel, or just want to hear more material that I talk about, please subscribe and stay tuned. Thank you for joining me.